Welcome everyone to this webinar today on liquid biopsy-based biomarker for cancer detection and monitoring. And I'm delighted to welcome our two renowned speakers, Dr. Klaus Pantel and Dr. Alain Thierry, professor actually, sorry for both, who will present you different perspective on liquid biopsy-based biomarker. And as an introduction, I think we are now living in a fascinating decades where there is a lot of ongoing active research projects and trials that will probably lead to more liquid biopsy biomarkers that will invade the clinical market and that will be fully integrated into healthcare system. So it is my pleasure to introduce our first speaker, Professor Klaus Pantel, Director of the Institute of Tumor Biology at the University of Medical Center Hamburg, Germany. Professor Pantel is the chairperson of this institute. And a bit of background: after graduated and a postdoctoral period in the USA, Professor Pantel performed research at the Institute of Immunology at the University of Munich for more than 10 years. And Professor Pantel is really known for his work in the field of cancer micrometastasis, but also circulating tumor cells and circulating nucleic acid, as reflected in more than 600 publications in excellent high-ranking biomedical and scientific journals. Professor Pantel was awarded with many awards, such as WCR Outstanding Investigator Awards in 2010 and many others. And Professor Pantel is also the founder and chairperson of the European Liquid Biopsy Society. In his presentation, Professor Pantel will discuss the conceptual framework of circulating tumor cell and circulating tumor DNA assay, and point out current challenges which might structure this dynamic field of translational cancer research. Professor Pantel, the floor is yours.
Thank you very much, Florence, for this very kind introduction. It's really a pleasure to give a short presentation today here at this webinar. And I think that I will, in the next 15 minutes, discuss very briefly the liquid biopsy field, how it comes from discovery to clinical implementation. These are my financial relationships. The concept of liquid biopsy was introduced in 2010 by Katrina Lix-Panabir and me, and we have really focused on circulating tumor cells first, but it has been quickly adopted to circulating tumor DNA and other analytes that are released by tumor cells, and it also now includes the host cells. The tissue biopsy is the standard method in oncology, but it's invasive and some locations are difficult to access. Even if you get a single biopsy, it may not represent other metastasis lesions in the patient. So the blood is a pool of tumor cells and tumor cell products released from all locations and may give more comprehensive information. Moreover, you can take sequential blood samples over time to real-time monitor tumor evolution and therapy response in individual cancer patients. So the vision is to get more comprehensive and real-time information by the analysis of blood or other fluids. Liquid biopsy and tracking cancer liquid biopsies was highlighted by Diana Romero in the latest Nature Milestones, and this was really great to see that it has seen such importance in the field. Where are the tumor products and cells coming from? In the beginning of the disease, the primary tumor is the major source. Then later lymph node metastases contribute, and in advanced stage patients, metastases are the key source of tumor cell DNA and tumor cell in the blood. The blood is also, of course, a key fluid, and I will focus on that today, but there are also other fluid like the cerebrospinal fluid or the urine or sputum that can be analyzed with liquid biopsy technologies. In the blood, we have the cellular fraction, and particularly the circulating tumor cells, but also the circulating host cells that can be analyzed. And we have in the plasma or serum the circulating DNA, but also other products like extracellular vesicles, proteins, RNAs, or tumor-educated platelets. The ctDNA is certainly a field of great interest for us for many years. And the circulating tumor DNA is mainly released from apoptotic tumor cells, but there's also active secretion through the extracellular vesicles. And I'm sure that Professor Thierry will focus on that more than I will do it today in my talk. But just to let you know that we have now very specific methods to detect minute amounts of tumor-derived DNA in the blood. And the concentration of the ctDNA depends largely on the stage. In advanced metastatic stage, you may have 1% or more of DNA in the blood, but in non-metastatic stages, you have less than 1% DNA derived from the tumor, diluted in the sea of normal DNA. And if you apply very sensitive methods, you have to be sure that you do not detect aberrations that are, for example, associated with age, such as the mutations in the leukocytes. Besides the ctDNA, the CTCs allow the analysis of viable tumor cells at the DNA, RNA, and protein level. And we have also developed functional models, such as the one that is quoted here on the slide, that allow now a more in-depth insight into the biology of tumors and also allow drug testing. So the CTC research, I think, can contribute to an understanding of tumor biology. We can address all the different features of tumor biology, in particularly also maybe discriminate of indolent versus aggressive cancer. And we have just published perspective articles on that in Nature Reviews Clinical Oncology. In order to bring really the wonderful publications into some benefit for patients and introduce it into the clinic, we have really established the European Liquid Biopsy Society Consortium, which consists now of more than 70 institutions from academia and industry. We have different working groups where we bring together the clinical working groups and the technology working groups. And we also have a regulatory working group, which will help to really get these assays reimbursed. And if you're interested in our consortium, please drop me or my project manager, Claudia Koch, a mail. We have been also involved in their guidelines, and these are the last ESMO guidelines published in Annals of Oncology. If you want to go through that in more detail, you see what are the current recommendations, and you will be, I think, told that in advanced cancer patients, we are already having very good assays that are sensitive enough to pick up druggable mutations if there is no tissue available. So what are the clinical applications? It's early diagnosis of cancer. It's also response and follow-up of patients to see who will relapse, getting information on tumor evolution, particularly resistance to treatment, and the detection of minimal residual disease. Let me focus on early detection of cancer first. We have just initiated a new consortium, which is focused on the early detection of pancreatic cancer. It's called PANCATE. It's a very aggressive disease, and we have today only very few patients that are detected at early stages, and we want to increase this incidence. So we are 17 partners from 8 countries, and I have the pleasure to coordinate this network together with Matthias Löhr from Karolinska Institute in Stockholm. We have different work packages. What is really nice is that we have large biobanks of blood samples from patients at early stage or even at risk to develop pancreatic cancer. We have experts on every analyte that is liquid biopsy related in the blood, from ctDNA to CTCs to proteins and metabolites. And we have a very nice, I think, bioinformatics group that hopefully brings that all together to find a classifier that can identify early stage pancreatic cancer. And in addition to these laboratory groups, we also have experts on health economic evaluation and patient organizations that will help us for the implementation into the clinic. And this is a group here from Hamburg. You see it's a translational research group. We have the liquid biopsy tumor biology group. You have the clinical colleagues, and you have the colleagues from the health economy research. The second application is early detection of minimal residual disease. So we are probably today in the good situation that with a liquid biopsy analysis, we can detect relapse earlier than the imaging shows. And this gives an opportunity actually to design new clinical trials. And we have called these post-adjuvant clinical trials a few years ago in NRCO together with Stan Hayes. So here we have a second large European consortium that started in May this year. This is actually a public-private EU-funded consortium with co-coordinator Klaus Andersen from Aarhus University and the company FPIA lead from Maria Carizades from BMS. Here we have the following objectives. We want to create reference standards and benchmark ctDNA for MRD detection, which really requires very sensitive assays. We want to do robust clinical validation of the DNA-based MRD in colorectal, pancreatic, and lung cancer as model tumors. And then we want to integrate MRD as a decision tool to guide treatment selection and develop future clinical trials in that space. And we are altogether 22 partners from industry and academia to reach this goal. The last area I would like to emphasize is real liquid biopsy in the field of immunotherapy, which has changed really oncology in the last years, and particularly in non-small cell lung cancer and melanoma treatment, but also other tumor types like bladder cancer and others. So what we're doing here, we are monitoring ctDNA and CTC concentration. We characterize these analytes. And we have also shown that some of the host factors, such as the coagulation system, could play a role in the response to immunotherapy. So we look both at the tumor and the host response. And we are now very happy that we can do that in a collaboration of an international network with Tatsuko Honyu, who is creating this network now. And as you can see, it's fantastic for us to collaborate in that context with Tatsuko Honyu, who received the Nobel Prize in 2018. Here, for example, and just to give you an idea of what we are planning to do, we have a very tight collaboration with the dermato-oncology group, which is here shown a lot of young, excited, and motivated people. And here we have an adjuvant cohort of patients receiving a double immunocheckpoint inhibition, and we have a metastatic cohort. We have a multi-center tumor bank, very dense collections of blood samples. And we have more than 10,000 blood specimens now in that biobank. And we look at the blood on various aspects, DNA, tumor cells, and proteins, and many other analytes. This is just one slide from our unpublished DNA analysis. We have here done a very intense analysis of circulating DNA with a method that goes down to 1 parts per million. And it also has the opportunity to give quite a range of information on different genes, as you see on the left side. And the ctDNA clearance in this pilot study predicted also patient survival, which I think was nice, and we will go on with this evaluation. I think what was also interesting to see is it's not only the tumor that is important for the response to immunotherapy, it's also the host. Here we could show that from Willebrand factor concentration, which is a representative of the coagulation system, was connected to primary resistance and was also in that context a prognostic factor. So in conclusion, early detection of primary cancer, MRD, early relapse requires very sensitive assays. The assays need to be validated and harmonized, and our ALPS network will help with that. The assays can provide clinically relevant information. Combination of different biomarkers may lead to higher accuracy. The resistance to immunotherapy may depend on both tumor and the host response. And we need, of course, interventional studies to demonstrate the clinical utility of liquid biopsy. And we are also in, I think, contact with the EORTC RESIST group to see how we can really implement ctDNA and maybe also CTC measurements in a new RESIST therapy response classification system. At the end, I would like to thank the people here at the UKE in Hamburg, which is a collaborative group of research institutes and clinics treating cancer patients. And I would like to thank the people in my institute, which consists of 60 colleagues from 15 different countries, a bit like United Nations. And a big thank you also to the funding organization, in particularly the ERC, but also the EU IMI, and EU Transcan organizations that has helped us with funding over the years, and the German funding organizations such as the DFG, BMBF, or Deutsche Krebshilfe. Thank you very much for your attention.
Thank you very much, Professor Pantel, for this very insightful presentation. There are many things, I think, to be discussed after with the participants. And now I would like to introduce Professor Alain Thierry, who has a strong expertise in research focusing on the diagnostic capacity of circulating nucleic acid, also called circulating DNA, and on the development of methods to support personalized medicine. His team has developed a preclinical model to quantify the different origin of circulating DNA and has demonstrated the importance of the detection of the short circulating DNA fragment for high specificity and high sensitivity analysis of this type of fragment. So Professor Thierry is working as a director of research at the Institute of Cancer Research in Montpellier in France, where he's coordinated the first prospective blinded multi-center study to offer clinical validation of plasma analyses which detect mutation in cancer patients. And more recently, his team was the first to demonstrate the clinical utility of using circulating DNA analysis for patients with metastatic colorectal cancer. His research also focuses on the other potential uses of circulating DNA. And his team has also led basic research program on circulating DNA structures and functions. They has notably demonstrated that most circulating DNA derive from neutrophil extracellular traps, which is pretty new. The team also discovered the presence in blood of circulating functional cell-free mitochondria. Finally, they currently coordinate more than 10 clinical trials, partly funded from European grant, but 3 basic research grants. And Professor Thierry is also the principal founder of the DIA-DX company set up in Montpellier, which is dedicated to providing liquid biopsy solution in oncology. So Professor Thierry will review the origins and the structure of circulating DNA in oncology and present you lessons learned from recent study and emerging approaches. Thank you very much. Professor Thierry, the floor is yours.
Thank you, Florence, for this very nice introduction. And thank you, Klaus, for this great work you are doing for a long time, first on circulating tumor cells and now on circulating DNA. That was a very nice presentation, and this would shorter mind and focusing more in new things. So thank you very much. And following this presentation, this introduction, I'd like to mention that the nomenclature of liquid biopsy is only made in the field of cancer, because there is opposition between tumor biopsy and liquid biopsy. So in regards to circulating tumor DNA, this field is also on scrutiny in sepsis, transplanted patients, and prenatal diagnosis. So we don't know that much about the proportion of the mechanism of release. And I'm going to present some new discovery at the end of the presentation. So what are the types of nucleic acids that we can find in blood, DNA, RNA, microRNA? And what it is interesting for sure is that these DNA, RNA, and microRNA can have the genetic and the epigenetic alteration of the cells they are coming from. So it is important to know the distinct cellular origins of circulating DNA, because this is one of the challenges of the circulating DNA in oncology, because circulating DNA from malignant cells are obviously with alteration, genetic alteration. But DNA, circulating DNA, are coming also from the tumor, but from non-malignant cells. So from the tumor, you have circulating DNA from malignant cells and non-malignant cells upon the progression of the tumor. But you have to know that everybody has circulating DNA floating in the circulation. And this is a question of concentration. There are around 5 nanograms per ml in every healthy individuals, but in cancer patient, it can be from 5 to 500 nanograms per ml. So knowing this challenge, and also to know what are the origin of circulating DNA, it is important to mention that there are between 50 and 1,000 times more genome equivalents in circulating DNA as compared to CTC. Then circulating DNA do not derive from CTC, and circulating DNA has better and higher analytical signals. Our work on circulating DNA analysis in oncology, as I said earlier, there is a opposition between tumor tissue analysis and plasma analysis, tumor biopsy and liquid biopsy. And plasma analysis has interesting characteristic because it is cost-effective, rapid, non-invasive, non-painful, easy, come from easy blood sampling, and it has a global evaluation of the genetic alterations, and also it allows serial analysis of a cancer patient. And what it is important that it takes account of the tumor heterogeneity of the tumor, because as you know, there is a intratumoral, intertumoral, and temporal heterogeneity of the tumor. So the circulating DNA take account of all the three heterogeneity types. So in terms of the methodological approaches, there are different approaches. That single locus assay, meaning essentially qPCR, or sequencing. And obviously, those methods provide different sensitivity. And upon the objectives of the use of the circulating DNA, we are using those different methods. What was important that we revealed 15 years ago that circulating DNA is highly fragmented. And this was important for reliable and accurate detection when designing target DNA sequences for pre-amplification or direct testing by qPCR. So we design a method, and with this method, we participate to the first interest of circulating DNA in oncology, meaning for targeted therapy, for selecting targeted therapy. We, as mentioned, Florence, we demonstrate the first clinical validation of the circulating DNA in oncology, and also the first assay on on the clinical utility. We found some interesting observation about prognosis, and also about following the heterogeneity, the clonal heterogeneity of the tumor. So quickly about the assay on the clinical utility, what we found is that in plasma, we observe better proportion of mutations. So it's more sensitive than the tissue analysis. And also we found that there is a co-occurring mutations. And through the circulating DNA, now co-occurring mutations are established. And 15 years ago, we believed that mutations were mutually exclusive. We evaluate the technical and clinical explanation of the level of discordance between tumor and plasma analysis. And the more important is the delay between tumor tissue and blood drawing. Very important to mention, as I introduced before, is the huge variation of the proportion of circulating DNA coming from malignant cells, meaning with mutation, among the patients having the same RECIST criteria. So in our second clinical trial, we found that the variation is ranging from 0.004 to 100%. We observed that the concentration of circulating DNA without bearing mutation, the percentage of those DNA and also a degree of index of fragmentation may have a prognostic value. So, as say beautifully Klaus, there are different potentials for circulating DNA, but we have to separate the quantitative analysis and the qualitative analysis, meaning more specifically genomic analysis. So, as said by Klaus, there are different potentials, and we are working in all those potentials in our lab. And we are, in fact, example what it is doing in many laboratories now, starting from molecular profiling and prognostication, detection of minimal residual disease, monitoring of the response, the monitoring of the clonal evolution, and also early cancer detection or screening. So just as an example, here you have illustration of serial analysis of metastatic colorectal patients after therapy. And as you can see here, you have elevation of total concentration of circulating DNA, which is in fact parallel to presence of different mutations. And as you can see that mutations can be different and co-occurring. Observation of Dr. Bardelli is the stop and go option for targeted therapy. In fact, sometimes you don't have, for example, for metastatic colorectal cancer, the detection of the mutation. So you are able to use targeted therapy, and then you can see elevation of circulating DNA, and you can stop the targeted therapy and go back to classical chemotherapy, and then reusing targeted therapy. So just to mention that we finished the inclusion and we start to analyze the data. of the first global interventional study where the analysis of the RAS-BRAF mutations for treating metastatic colorectal cancer patients with targeted therapy is carried out by analysis of circulating DNA. We also use a circulating DNA for observing the elevation of the tumor burden in patients after the COVID-associated lockdown because of lower diagnosis proportion in the global population. Now I'm going back to the basic studies, especially on the circulating DNA fragmentation origins. So as I mentioned earlier, we found that circulating DNA is highly fragmented, and this fragmentation is based upon the topology of the DNA, which is associated principally with histones, so in the mononucleosome. And we observe here, for example, that more than 90% of the circulating DNA are associated with mononucleosomes. And what it is striking, as you can observe here, in plasma of 17 different healthy individuals, you have exactly the same fragmentation profile. And as you can see here, you have a very a scale of base pair. So this is a very intriguing how this is reproducible. And these characteristics allow us to make a difference between healthy individual here in blue and cancer patients here in black. And you can see that the fragment profile of the cancer patient is different. So we can use fragmentomics to develop cancer screening tests. So there are different options using fragmentomics because we can work on the fragments, we can work on the nucleosome occupancy and the nucleosome depletion erosion. Then the use of circulating DNA is very rich in terms of the information we can get, especially in the oncology field, because the goal is to differentiate healthy individual and cancer patients. More on the origin, structure, and function of the circulating DNA in oncology. First, the paradigm was that circulating DNA was originating from apoptosis or necrosis, but I never believed on that. And our hypothesis is that chromatin is degrading from big filaments to mononucleosomes, big fiber. And our hypothesis is that nets is a possible origin of circulating DNA. Nets are neutrophil extracellular traps produced by neutrophil when there is, for example, infection. And they are intended to kill bacteria and eliminate them. But nets are sometimes overproduced and they are implicated in various diseases. And they are also implicated in tumor growth. So there is an increase of concentration of circulating DNA in blood in all of these diseases. And our hypothesis is that nets are one of the major sources of circulating DNA. So we carry out a very large investigation in vitro, ex vivo, and in vivo. And we found that neutrophil extracellular traps have autocatabolic activity and produce mononucleosome-associated circulating DNA. And this is in line with the beautiful work of Dr. Moss and Yuval Dar from Jerusalem, that found that with deconvolution from methylation atlas, we can determine the proportion of the origin of the circulating DNA cells. And in this work, it is clear that neutrophil is one of the principal origin of circulating DNA in healthy individuals. So we work on NETs and cancer, and we observe that the marker of circulating NETs and circulating DNA are significatively higher in metastatic colorectal cancer patients as compared to healthy individuals. And we found that all those markers are highly associated, and they have diagnostic capacity. And at the end, so we postulated that there is an association of neutrophil extracellular traps with the production of circulating DNA in patients with colorectal cancer. And we found the same thing in the COVID-19 patients, because in fact, the acute phase of COVID patients is associated with elevated NETs, and we found that those NETs are also elevated in long COVID patients. Pass by, and the diagnostic capacity of those markers are very high in COVID. About early detection of cancer, we found that circulating mitochondrial DNA, which is this second type of DNA you have in blood and in the circulation. So we observed that these circulating mitochondrial DNA may have capacity in differentiating healthy individual and cancer patients. We realized a clinical trial on more than 1,500 individuals, and we used the artificial intelligence or machine learning for selecting the best combination, and we obtained something interesting, but it is not maybe enough. And we plan that different analyte from circulating DNA may be combined in a test for screening cancer individuals. We lastly compare the structure and the topology of plasma extracted circulating nuclear and mitochondrial cell-free DNA, and we observe that the great majority of circulating DNA from the nucleus, from chromatin, are associated with mononucleosomes. There are some in vesicles, and we observe within the same method the different topology of circulating mitochondrial DNA, and we observe that the majority of the circulating, and more than the majority, because it's more than 75%, that circulating DNA from mitochondria are in fact in form of free mitochondria circulating in blood. And we presented recently a discovery about the presence of circulating cell-free respiratory competent mitochondria in blood, and this was of great interest for diagnostics and therapeutic objectives. So I thank everybody, and especially my team members, and I will be happy with Klaus to answer to your questions.
Thanks a lot, Professor Thierry, for this very comprehensive presentation on and especially on the new concept of origin of circulating DNA, which is very innovative. So now, if you can stop sharing your presentation, please.
Sure.
So in these two previous presentations given by Professor Pantel and Professor Thierry, we heard more about blood-based biomarkers, and I would like to give you an overview of what we are doing at IARC to use urine-based biomarker as a non-invasive approach for the detection of bladder cancer and the monitoring of bladder cancer. So let me share my slide with you. You can see them.
Yes, perfect.
Perfect. Okay. So just a bit of background in terms of bladder cancer. Few figures. Bladder cancer is still a pretty common cancer because worldwide is the 10th most common cancer, and it's still the 13th deadliest cancer in the world, according to our IARC GloboCon data. The risk factors for bladder cancer mainly include tobacco smoking, because tobacco smoking is responsible for more than 50% of bladder cancer, at least in the Western countries and developed countries. But there are also other risk factors that can include past radiation exposure, chronic inflammation of the bladder, and also something which is probably a bit underestimated is occupational exposure from chemical industries. So these risk factors are responsible for the urothelial carcinoma that are actually representing more than 90% of bladder cancer in developed countries, as you can see here. It's different in Africa and in the Middle East, where still the parasite infection of schizostoma, which is a parasite found in some endemic region in Africa and Middle East that contaminate a lake. And this parasite is responsible for the development of a particular form of bladder cancer, a squamous cell carcinoma of the bladder. So in terms of diagnosis, in fact, when there are symptoms, the main symptoms include blood in the urine for, let's say, majority of the patient, 80% of the patient present with hematuria. But it could be also like a bladder pain that the patient feels. So in the presence of these symptoms, the patient is referred to a urologist that will propose several tests. As you can see, there is urine cytology, but there is also urine-based biomarker that exist, in fact, but they are listed here, the one approved by the FDA, but they are not really clinically used in routine practice. And the reason is that they were commercialized, but they were showing afterwards lack of performance consistency for the result. Urine cytology, which is based on the detection of malignant cell from a urine sample, will lack of still lacking of sensitivity, especially for low-grade tumors. So in the end, the main technology still used for the bladder diagnosis is the cytoscopy, which is not very pleasant because it's invasive. It consists of the insertion of a camera into the bladder via the urethra to visualize the inside of the bladder. Plus, it's pretty expensive. So we need more research on bladder cancer biomarkers. And this came to us about 10 years ago now, with the first discovery of two mutations in the promoter of the telomerase reverse transcriptase genes, also called TERT. As you can see here, there is 2 hotspot mutations that were initially described in melanoma cells, and afterwards screened in more than 1,000 tumors of 60 different types from the Johns Hopkins Institute. And they were found to be highly prevalent in glioblastoma, some sarcomas, but also, as you can see here, in bladder cancer and carcinoma of the upper urinary tract system, where they have been identified between 60 to 80%. So we thought this represents a really nice candidate biomarkers, like very simple, few mutations that are really specific to a genomic region. And in bladder cancer, as you know, the urine is of course in direct contact of the urothelium, and when there is a tumor, it's also the case here from this figure. I adapted these figures where you can see that some intact cells here from the bladder will desquamate into the urine, which could be a type of source of DNA that we focus on, but there will be also release of cell-free DNA, as our speaker talked about, also in the urine. So basically, when you take a urine sample from a patient and you centrifuge it, you will have in the urine supernatant the cell-free DNA fraction, which will contain a mix basically of tumor cells, but also, of course, a lot from the normal DNA of urothelium. It's the same for the urine pellet the urine pellet, which will contain the exfoliated cells, where we can extract genomic DNA from this cell, but again, that will be a mix of normal cell and tumor cells. So here in the lab at IARC, what we've done is that we've developed 2 assays to detect these 2 main mutations that we call the C2280 and the C250, but also 2 other rare ones that are shown here. So we basically first developed an NGS-based assay that we call Euromutert, that we associated with a homemade algorithm for the detection of very low level of mutation, because as I explained you can have a very low allelic fraction, so you want to have a good analytical sensitivity of the methodology, like Klaus and Alain presented. The result, well, I'm going to show the result, and it was a very nice technology, but we had also in mind the clinical potential of the assay. And because of that, we thought, okay, it will be much more simpler to adapt the assay to a droplet digital PCR. The reason is that you can get the result in a few hours time. It's really straightforward. You don't need bioinformatics analysis, and it's pretty cheap too. So we did both. In parallel to this development, we collaborated with a local clinic here, the Infirmerie Protestante de Lyon, to include about 100 bladder cancer cases and about the same number of hospital-based control, for which we prospectively collected urine sample, of course, blood sample, saliva, and also tumor tissue for the cases. And I'm going to give you really briefly the result of this study, just maybe for the urine. When we combine, in fact, the analysis of both urine supernatant, so cell-free DNA and cell DNA, we reach a sensitivity of 87% of detection using our mutation in the TERT promoter gene, for a specificity which is reaching 95%. When we stratified also the result according to the risk categories that we got in our study, low risk, high risk, but also muscle invasive tumors, you can see here, we saw that first, TERT promoter mutation were found in every category, so they were equally performant. And in low-grade tumors compared to cytology, it of course outperformed this method. So we were excited about this result, and we asked ourselves, can we detect our marker in preclinical samples? So basically years prior to diagnosis. But to do so, we had to see, to work with a prospective cohort study where they have basically collected urine sample at enrollment. And for this, we historically at IARC, we collaborated for a long time with the Golestan cohort study that actually included more than 50,000 participants in the early 2000s. And they followed these healthy people for more than 15 years now. And initially it was developed to study esophageal cancer, but they were very nicely organized and they record any kind of cancer and actually other disease. So we asked the PI, Dr. Malek Sadeh, how many bladder cancer cases did you have in your cohort with urine collection at enrollment? So they had 40. To which we match one case to 4 controls, matched for date of birth, sex, date of urine collection, and residence. And we applied our assay in a stringent manner because we applied the NGS-based approach first, and we confirmed the variant identified by this approach by droplet digital PCR. And what we showed is that we could detect the marker for about half of the cases with a perfect specificity, so no detection in the one that did not develop the bladder cancer over time. But the most striking thing was that we could detect the marker up to 10 years prior to diagnosis, as illustrated here on this figure that shows, in fact, the allelic fraction, so the mutation load, basically, of our mutation, according to the time of diagnosis in the x-axis. So what you see is that the closer you get to the diagnosis, the higher is the allelic fraction, which basically makes sense. So what does it tell us? It tells us that our marker is a very early event in the process of in the carcinogenesis process. So what are we doing now in terms of monitoring? Because bladder cancer is actually the most recurrent disease in terms of malignancy. They have the highest rate of recurrence. So the marker could be also interested to monitor this recurrence, because patient diagnosed with bladder cancer, if it's a high risk, for example, they will be subjected to cystoscopy every 3 or 6 months, depending on the risk they have. So we followed the French cohort and collected urine sample at every medical follow-up, as you can see here, for at least 5 years for about 50 patients. And we applied the TERB marker and compare with urine cytology and cytoscopy result when available. So we are now doing that. It's ongoing. We stopped the follow-up, but it's just to show you some preliminary result of a specific patient here that was included in 2016, for which the marker was positive in both the tumor and the urine pellet. We don't have the data for the supernatant, but after 1 month after surgery, you can see, and as Klaus was discussing also, that the marker is still positive here, showing probably that there is a minimal residual disease already here at this step, 1 month after surgery. Then we monitor, you can see the dynamic evolution of the marker that really show, in fact, a rapid increase after 4 months after inclusion and 7 months, where cytoscopy, sorry, showed positive result, and the recurrence was shown here. And again, after the recurrence, a rapid drop of the marker. Another interesting thing is that we say, okay, but if there is positive marker at first at diagnosis, and then can we avoid repetitive cytoscopy in TERT negative patient as illustrated here? So this patient was treated with chemotherapy after surgery, and you see for a long time was completely negative for the marker, and also normal at cytoscopy. So really, you know, for comfort and also release a bit of the cost burden for the healthcare system, we think this could be interested also in this way. So as a wrap-up, the potential clinical application of Urine-based is highlighted here is IARC summary evidence brief that we wrote last year. You can download it from this website, from the clinical application of the marker. Now that we know that our marker is present years prior to diagnosis, of course, the idea would be to go for intervention study or screening study to show that in people with some risk factor, also presenting with a microhematuria, we could detect TERT and they could be referred to urologists afterwards. And of course, everything that I showed you here for the monitoring of bladder cancer. And I thank you for your attention. There is also a lot of people involved in this study. I don't have the time to thank them all, but I want to say special thanks to our postdoc that contributed to the two studies that I showed, Patrice Avogbe, Dr. Ismail Hossein, and Dr. Madi Sheikh. Thank you for your attention. And I will unshare my slide. And now I think open to discussion, open the floor to discussion. So if you have questions, I've seen that some people were typing in the chat, please use the Q&A. And there is a question from Céline de Ropp. Are there any recommendations for methylation analysis on liquid biopsies for diagnosis or follow-up in oncology? Who wants to take this, Professor Portel?
Yeah, I'm happy to answer that. I think that we are working on that. That's actually a very good question. And I think that the methylation DNA field has technically changed over the last years, and I think that such recommendations could be extremely helpful, technical recommendations, and then also recommendations on the potential use cases in the clinic. And I think Alain Thierry has also mentioned some of the applications, from Yuval Dor, the excellent work, which I think is also part of our PUNC8 consortium. So I'm not aware that there are already recommendations published, but maybe Alain, you have more information.
Yes, you're right, Klaus. Methylation is a very important field now in circulating DNA because recent demonstration of the atlas and of the cell origin of circulating DNA from the deconvolution of sequencing of circulating DNA through the methylations allow enormous possibilities. For example, to distinguish between cancer patients and healthy individuals. And also, it might have importance to know what are the tissue that are in the body degraded or deteriorated or altered by other pathology than cancer. For example, last work of Yuval Dor showed that when the liver is altered, you can observe it accurately, and this is of importance not only for cancer but for many different pathology or normal situations.
Absolutely, and I think we have published a paper together with our neurosurgeons looking in patients with glioma on methylated DNA in the EV fraction, which was very interesting work of Frank Ricklefs and colleagues. And he could really show very nicely that he can molecularly, distinguish the different classes of glioblastomas using that information, which I think was very helpful to see. So even for brain tumors, you can get enough information out of the blood analysis, which is very promising, I would say.
And if I may add, also, I think we are we are still in a lot of assays, in fact, in the validation phase, and just starting with some additional intervention work. And I guess the regulation will come after this, the result. I'm thinking of the multi early detection test that, for example, are now being investigated by Grail, for instance, and other companies. But the phase is that, so there is another question about, so is ELBS working with regulatory agency to define the requirement to validate the use of liquid biopsy? So it's even prior in the clinic. And if yes, can you develop this requirement and where it stands today in that regard?
Yeah, we have actually established a new working group, which we call regulatory working group, which exactly does this interaction, and it's now taking really pace. What it makes a bit more complicated in Europe is that each country has different rules, right? I mean, in France, I think it's more centralized. In Germany, it's more federated system, and we have all kinds of different healthcare companies. So, but with ELBS, we are just bringing all this information now in our meetings together, and we are also developing, I think, a roadmap how to add and bring these technologies into the reimbursement scheme, because the reimbursement is, of course, key, you know. I mean, if you want to do, let's say, these analyses at a larger scale and also outside of clinical trials, you have to make sure that the reimbursement is also taking place, right? And not just for a few people who pay privately for that.
Yes, definitely. The question of reimbursement of such NGS tests, for instance, you know, like for the moment, it's still pretty costly, because I think it's around $1,000, for instance, the methylation test. Or so, for example, for early detection, it will be extremely hard to convince the regulatory agencies that it's worthwhile and cost effective, unless the cost reduced dramatically. But the first step is to validate that it's working.
Yeah, I think the first is always to make sure it's really working. And then, I mean, if a test is being used many times, and then I'm always optimistic that there will be, let's say, technology advances maybe to reduce the cost, you know. I mean, think of the costs of sequencing 10 years ago or so. I mean, it cost you a fortune, right? I mean, today you can get it for maybe $1,000 or whatever, right? So maybe, I mean, we should not be stopping things because we think the costs are too high, but rather, you know, look at the clinical data, whether it makes sense, and also whether the patients will profit from it. And you have also shown nice examples with the bladder. You know, it's much nicer to take a blood sample than make a bladder cystoscopy, right? So I guess that that the patients and the individuals in the countries should really profit from these technologies, I would say.
I would like to add from Klaus' arguments that we have to distinguish between cancer screening and cancer patient management care. And as say Klaus, cancer management care is very costly, and circulating DNA, NGS, and other costly analyses are worthwhile because it really make it can really make a difference. Okay. The problem with cancer screening is that you are using it for the whole population, and the cost here is of an issue. but I don't think that the cost will be an issue for cancer management care.
Yeah, I think
less.
I fully agree with you. I think these are two totally different, let's say, applications, right? And like in my country, in Germany, we actually reimburse, I mean, things for patient care, you know, not so much for prevention, you know. So I think that it's also a totally different, I think, reimbursement scheme, whether you have an early test of cancer or an MCD test, or whether you have a test for cancer patients with a diagnosed tumor, you know, receiving some potential treatment. So I agree. These are two very different scenarios. Yeah.
Thank you. We have a question, a very specific question for Professor Thierry. How to consider early detection of cancer if there is a very heterogeneous detection rate by cancer type, let's say high detection rate in Hodgkin lymphoma? Is it explained mostly by circulating origin of NET, cancer with high percentage of inflammatory reaction in the stroma?
In fact, there are two questions in the
Yes.
in this one. In regards to early detection, I believe that we have to also to consider two scenarios. The pan cancer, which is a screening test for detecting all cancer types, or the specific cancers early detection. And this is after a visit with a physician. And if the physician has a question, he can use early detection from circulating DNA, but for a specific cancer types, okay, depending of the symptoms, okay. But for pan cancer, it will be much more difficult because, as said, the intervenant, the attendant, it's difficult to validate a test for all cancer, okay? So this is two scenarios. Second part of the question rely on the nets. And I believe that nets can participate to the diagnostic capacity of circulating DNA. But he is right that, in fact, nets are involved in inflammatory disease. And why nets are involved in cancer is because cancer is an inflammatory disease. So, and hopefully it's an inflammatory disease because, as you know, immune response is very useful to control the disease. So nets are really, for me, a marker of tumor progression, but you have to consider any other inflammation process, and is right. So you have to be cautious about this point.
Thank you, Professor Thierry. We have a question regarding TERT, where there are any changes on cytoscopy in preclinical patients with positive urine test, and if not, what should be the clinical approach in this case? Very good question, and I can answer that. You know, so in the validation phase of a biomarker, we basically, when we did the prospective cohort study with Iran, we are in phase 3, where you basically look into samples at enrollment, but of course you don't intervene. So you don't know if the urine sample that contained the positive marker will actually be positive at cytoscopy. The only way you can do that is through a screening trial, and this is what we would like to do, is that we take a high-risk population, or, for example, maybe easier to convince the ethical committee is that first to try it on people with hematuria, because anyway, people with hematuria will undergo cytoscopy. It's the guidelines, basically. So we could compare with TERT, prior to primary diagnosis, if it's worthwhile already. Then after do it the same with microhematuria and people with no symptom, but with risk factors, such as people that have occupational exposure. But this needs intervention studies. And we have a question on the role of minimal residual disease in lung cancer. Who wants to take this question? Lung cancer.
Well, I think that we have the lung cancer included in our new EU network guide MRD as one of the 3 tumor types, where we will do a follow-up in patients with stage 2 or 3 tumors, to see whether we can pick up the relapse earlier. That has been reported that you can pick up the relapse earlier, but we will compare different technologies. And we will also implement and test different DNA references. And that's also important. We probably also have to implement EQA reference standards, right? To make sure that if we have these sensitive tests, and we have a low concentration of DNA, that the test always works very nicely from day to day and week to week, And so that's also, I think, so we are doing that also in lung cancer. And we hope to develop that over the next years. Of course, in lung cancer, you also have the advantage that you have a lot of druggable mutations. So you can not only track the concentration and burden of, let's say, MRD, but you can also get very nicely qualitative information, what kind of mutation is coming up, and whether you have a drug that can target that. Yeah. Mm-hmm.
I can add to Klaus' arguments that for lung cancer, the question is relevant because many times the tumor volume is very low in lung. So in terms of the minimal or residual disease, it is important to get a sensitive assay to determine the mutations at the baseline. And it is difficult when the volume of the tumor at the start is low. And especially also the number of mutation is pretty wide, at the exception of the PIK3CA mutation and the KRAS mutation. And at this time, the work is more focused on the EGFR mutations. And we are working in France in a 10 million euro project on specifically this task. And we hope that in a few years or 5 years, we will get some interesting result. And I think it is important to get very sensitive technology. One of the options would be to get NGS assay at the baseline before surgery, and knowing the mutations, and then use a qPCR advanced assay, which is much more sensitive than NGS, to detect mutant circulating DNA and to achieve accurate minimal residual disease detection.
It's interesting because we have a question about qPCR that I think you can answer also because you worked a lot on qPCR also, the Professor Thierry. Is it possible to detect biomarkers from liquid biopsy using qPCR, even if we know that its sensitivity is low? It's exactly what you said, probably if you target a few.
No, this is the contrary. NGS assay, the conventional NGS assay has a sensitivity of 1%. The advanced NGS assay have a sensitivity of 0.1%. qPCR-based assay can have a sensitivity of 0.001%. So 100-fold more than NGS conventional or 10-fold as compared to NGS, advanced NGS protocol. The problem is that when you use qPCR, you are in a scenario when you have informed assay, information on the mutation. NGS is very useful when you don't know the mutations. And this is important. Or qPCR is very useful also when you are using targeted therapy because there are only few mutations of mutant genes that are involved in the actionable drugs. So qPCR can be also combined, as I said, Florence, you mentioned very well, that qPCR can be combined after NGS assay because it's very costly and you can have advanced NGS. And then for the serial analysis using qPCR, detecting the mutation that were detected from the NGS at the baseline.
Thank you. If we stay on the technical consideration, there is a question: is that what was the yield ratio from tumor DNA from urine supernatant versus pellet? So what we saw is that in the majority of the time, there is always more DNA coming from the exfoliated cells rather than in the supernatant fraction cell-free DNA. Probably makes sense. The only few exception, if I recall well now, is that we had few cases of carcinoma of the upper urinary tract, for which it was the opposite. So we had more cell-free DNA than really mutation in the cell pellet. And it just may be due to the fact that, in fact, it's quite logical that you may release less cells from the ureter, for instance, into the urine, but the cell-free DNA will be still be present. What we saw also is that we didn't find any logic basically between a fraction of TERT mutation, except for the high-risk category, where we saw that they were a bit higher in terms of mutation load than the other type of cancer. So either muscle invasive or low-grade tumor, for instance. We also have a question on breast cancer. Where was it? Breast cancer, I saw it. Yes. There are studies that correct the response to neoadjuvant chemotherapy for breast cancer and ctDNA level. Is there any relation between ctDNA levels and recurrence in a post-adjuvant setting?
Yeah, I think there are more and more data that are really coming up in this topic. We have also done quite a few studies in breast cancer. There's also the original work from, Jackie Shaw's group that has shown that she can detect ctDNA signals during the follow-up more than 700 days, up to more than 700 days before the imaging. So that would give quite a large window of opportunity, I would say, for post-adjuvant therapy. And we have set up a large multi-center randomized trial here in Germany. The lead PI is Wolfgang Janni from Ulm. It's called the SURVIVE study. And here we will include 1,750 breast cancer patients in the ctDNA screening arm and compare that with 1,750 patients that are, following the standard follow-up, right? And this is probably very important to really show, if we compare it to the standard way of follow-up, is it really reducing, let's say, relapse, or is it prolonging life, or is it improving life quality? So we probably always need to compare that really to the standard way of doing the follow-up to really convince, let's say, the clinical co-workers and the patients, of course, that we can not only detect something earlier, but that also will help them, right, in the future. Breast cancer is, of course, a very good model, because ER-positive breast cancer has this long latency period between early diagnosis and relapse. It can, 10 years or later, you can have a relapse. So it would be very important to tell the women whether they are safe or not, right? So absolutely. Mm-hmm.
Florence, unmute your microphone, please.
Excuse me. A related question regarding more legal issues in Europe is that, is there any reimbursement in Europe for MRD assays, only available for EVDR compliant assay, or does it extend to laboratory developed in-house test? No, not yet. Who wants to take this? I mean, for the moment,
I can start quickly. A brief answer is no. I'm not aware that there is any, of course, established MRD test in Europe. I know we had MRD tests, as far as I remember, one of the providers who got FDA approval for MRD, but in Europe, as far as I know, there is not such a test yet. But it's certainly our goal to develop such a test within our GUIDE MRD consortium, and then also see which tests, are in-house or not in-house tests, to compare them. In principle, I personally like more, let's say, decentralized testing, where special laboratories like the lab of Alain and others can really perform a test rather than just sending all the blood sample to one centralized facility. But that's because we are doing the tests, right? We like it more to really also be participating in this testing rather than just sending blood samples to a central facility. But I don't think that there are any of these MRD tests, to my knowledge, in Europe, be I think approved at present. Mm-hmm.
I agree with Klaus, and I like to add that in fact there are technological issues to standardize the method, but the demonstration is made that circulating DNA can detect minimal residual disease. But there is another issue, is the fact to standardize what we will do with it. So there are a different option, is that to reduce the intensity of adjuvant therapy, or to withdraw adjuvant therapy for certain patients. So, and this degree of intensity, or yes or no, it's not yet defined. So I think for the time being, this is also an issue that impede the implementation of circulating DNA in the MRD detection.
yes. And
I think we have, I should also add, it has been nicely also outlined in the ESMO recommendations that were published last year, I think it's still pretty up to date. Where are we with the MRD to date? Thank you.
And there is a related question about urine test with using NGS. And yes, there is some which are developed in UK, but also in the US. And I know that the company in the US, they also try to validate it for MRD and recurrence, and they are trying to bring the proof with several studies that it could be cost effective and taking part of their medical healthcare system and reimbursed. So there are some initiative, but still only.
I like to add to Florence's answer that urine test is very interesting, but there is a drawback, is that in fact, in contrast to circulating marker, that where there is homeostasis in blood, there is a difficulty in determining quantitative analysis. And I think that that many studies are focusing on qualitative analysis, but for example, detecting circulating DNA from malignant cells. But I think that it is interesting to have some quantitative information, such as the quantity of circulating DNA mutant and the quantity of the total circulating DNA. So, and this is very difficult when using other biological source than blood.
Yes, definitely. I was referring to a test for bladder. For now, there is only for bladder. Otherwise, it's totally true. What you just said. I think it's much more complicated with urine for other organ sites. And I don't know if we have time for a last question, which is also interesting. Last question, Dominique. Why choose circulating DNA rather than RNA? But there are a lot of studies also on microRNA and other type, you know, like there is another question about exosome. I think now the idea is with all the technology that we have, we can focus on the multi-analyte approach and try to see what is the best. Do you want to add?
Yes, I can mention that one of the difficulty with RNA is that RNA is more sensitive to degradation, and RNA is less protected in blood. So in regards to quantitative issue, this is also a problem. But also we have to mention that there are different kind of RNA that you can detect in blood. You have messenger RNA, you have long RNA, or you have microRNA. In regards to the number of study, microRNA are subject for many investigation, and some panels of microRNA around 12, 15, or less than 10 of microRNA were defined and followed during cancer progression. There are a lot of patents on that. The problem so far, they are not demonstration of their use, but it is possible to think that in the future microRNA panels can be used. And one of the difficulty with messenger RNA are the detection, as I said, but I think it could be very interesting. And we can also mention something that we didn't mention today is transcriptomics. We can use transcriptomics from circulating DNA, and that could be very interesting because we can retrotranscribe RNA to DNA and make sequencing and determine qualitatively the RNA. So in our team, we have a project on the detection of messenger RNA, we have already detect some specific messenger RNA of colorectal cancer. So more is coming with RNA, but the question is more on the capacity of detecting RNA, which are more fragile than DNA.
I mean, one way to get around this is, of course, to look at the RNA, which is protected in the EVs, right? So that could be also interesting. So the EVs are also super interesting liquid biopsy biomarker, and they are also interesting proteins in the EVs. We didn't touch on that today. So as Ellen said, there's more to come. Stay tuned. Huh?